This is Ashwin Khanna from second year MTech KA. I'm here to present my dissertation project with the title Reduction of Lung Adenocarcinoma using DNA methylation under the guidance of Ms. Geeta. Let's have a small introduction of what this my project is about. So lung cancer is a leading cause of cancer-related deaths all over the world, mainly due to the late-stage diagnosis. So early detection is critical, but current methods like CT scans and biopsies are either invasive nor effective, or not that effective for detecting the cancer at very early stage. So this creates a need for a non-invasive and reliable diagnostic approach. So DNA methylation offers a strong solution as it is an epigenetic modification that occurs early in the cancer development and remains stable over the time. So importantly, it can be detected from the blood samples, making it suitable for the early screening. So at the same time, transformer-based models are also highly effective for sequence data and capturing the contextual relationships. So in this work, we leverage the transformer-based deep learning to analyze the DNA and DNA methylation in a context-aware manner for the early lung cancer detection. So before we get into the project, so let me explain what exactly the DNA methylation is. So the DNA methylation is a genetic modification, meaning it regulates a gene expression without even changing the DNA sequence. So it mainly occurs at CpG site levels, where cytosine nucleotide is followed by the guanine, which is a chemical composition which presents the DNA. So the process carried out by the enzymes called DNA methyl transferase, so which add the methyl group like such as CS3, CS3 to the cytosine base, forming an 5 methyl cytosine. So when this methyl group is added, it can be a block translation factors for binding and cause the DNA structure to become more condensed. In the sense, it became more tight. So as a result, the gene becomes inactive or silenced. So in a normal cell, this mechanism helps to control the gene expression properly. But however, in cancer, this process becomes abnormal. For example, for tumor suppressor genes can be hypermethylated, which turns them off. So this removes a cell's ability to control the uncontrolled growth of cancer. At the same time, other regions may become hypomethylated, leading to the activation of harm genes. Hypomethylated is the undermethylation, and hypermethylated is overmethylation. So this is why we are using DNA methylation in order to see the presence of cancer. So although this DNA methylation data has a strong potential for cancer detection, existing computational approaches have several important limitations actually. The first is like the most models treat the CPG sites as an independent features, whereas like in reality, the CPG exhibits an interdependent behavior within themselves, especially within the nearby genomic regions. Ignoring this can lead to the loss of important biological information. So secondly, many approaches do not incorporate the DNA sequence context, even though the sequences plays an important role in the gene regulations and influence the methylation patterns. Thirdly, existing models struggles to capture the genome-wide interactions, particularly long-range dependencies across the different regions of genome. In addition, like traditional machine learning methods faces a challenge like with high dimensional methylation data, while deep learning models often lack interpretability, making them too difficult to trust the clinical settings actually. So because of the limitation, there is a currently less models that can jointly integrate the methylation signals and sequence information and biological context in a scalable and meaningful manner. So the primary objective is to develop the deep learning model for an accurate detection of lung cancer using a DNA methylation. To achieve this, the model is being designed to integrate the multiple layers of biological information such as methylation signals, genomic contacts, and DNA sequence information. So another key objective is to handle the high dimensionality of methylation data by performing the effective feature selection so that only the most relevant CPG sites are used for the model. So in addition, the model aims to capture the spatial and functional relationships between the CPG sites using the transformer-based architecture, allowing it to learn both local and the contextual interactions between the CPG sites. So finally, an important objective is to ensure the interpretability so that the model can identify the significant CPG sites and the biological relevant genes that are associated with the lung cancer. So this makes the system not only predictive but also clinically meaningful. So we did have quite a few literature surveys on the papers which is being currently published based on the DNA methylation, the cancer prediction, and the transformers model. So here are some papers which I have been put here to for the case study. So the first, starting with the first paper, which is on mutant method. So this work focused on predicting the CPG methylation status using a genomic sequence context. It uses the transformer-based architecture with KMR tokenizations, treating the DNA as a language. So the key strength of this model is an ability to learn the local sequence pattern effectively. But however, it is a limitation that it's not focused mainly on the sequence-based prediction, but I mean, it focuses only on sequence-based prediction, but it does not fully integrated genome-wide methylation interactions or classification tasks in order to predict presence of cancer. So second paper is on methyl profit. So this work addresses the issue of missing CPG sites in the methylation arrays. So it uses a gene guided bottleneck transform where the gene context cells and DSS are encoded into a compressed latent representation. So this is an effective for recovering the incomplete methylation data, but however it's being designed for imputation, not for direct cancer prediction or classification task. So this model and third paper is methylbird. So this model is focused on tumor classification based on the read level data, not the actual data set which has been taken from the biopsies. So it applies the bird-based architecture encoding the methylated and unmethylated states as a token and learning the spatial relationships between the CPG sites. So, but its strength is capturing the fine-grained methylation patterns, which is an in-depth way of seeing a CPG site. But however, it operates at the local fragment level and does not capture the long range or large scale genome-wide interactions between the CPG sites in along the sequence. So fourth paper is on hybrid GNN transformer model. So this work aims to capture the complex biological interactions across the multiple omics data, not only from the DNA methylation, it uses the RNA, it uses the histone modification data also. So it uses the dual branch architecture combining the graph neural network for the local interactions and transformers for the long range dependency. So while it improves the interpretability, captures the complex region, but it introduces high model complexity and it's not focused specifically on the methylation driven cancer prediction. So fifth paper is finally we have a deep learning based lung cancer classification. So this model performs classification on LUAD, which is the cancer lung adenocarcinoma, which is lung cancer we are testing, and LUAC is another subtype of the cancer using a methylation data. But it uses the supervised deep learning method, it's an MLP, so with a selected CPG's features. So while it achieves the classification, its limitation is that it treats the CPG site as an independent features, does not capture the contextual relationships or the biological interactions. So based on the papers we reviewed and several important research gaps can be identified actually. So first is that there is no major joint, like no joint modeling of DNA sequence and the methylation data. So most existing models focus only on sequence like transformer based genomic models or like no only they are only focusing on the methylation values but not together both with the context gene context data. So second is that there is a lack of gene scale interaction modeling. So many models capture only the local relationships between the CPG sites. but it's failed to learn the long range dependencies across the whole genome actually. So which is also biologically important. So third is like existing approaches like cancer specific modeling. So most models are designed general tasks like methylation prediction and imputation, but rather than focused on clinical applications such like cancer predictions or cancer classification. So another important innovation is like imitation is that like lack of interpretability. So many deep learning models behave as in black boxes, so making it difficult to identify which CPG sites or the gene, which kind of gene are actually contributing to the prediction, or actually plays a major role in the presence of cancer. So which is very critical in the medical application. So finally, there is no unified and clinically applicable framework that integrates a sequence information, methylation data, and biological. So let's dive into the project. I mean, the model which I'm working. So before that, let me see what is the data set being used. So let me say what is the data set being used. So for the study, DNA methylation is being used, as I told you before. So this data was collected from the two major publicly available repositories. One is GEO and another one is TGCK. So which are widely used for the cancer research purpose. So this data set consists of total of 1,614 samples, which is being extracted from 1,614 patients. So which this includes 1,119 of LUD samples and 495 of normal samples. And with that, we ensure that, I mean, we have the stage data with the LUD samples, ranges from stage 1 to stage 4. So I mean, this is a representation of a structured Bayesian work, this is an aggregated data where this data has been transformed from the actual raw data sets. So this each sample is represented as an structured JSON format, which allows to organize both clinical data and CPG level information efficiently. So at sample level, we store the sample ID, data sources, cancer label, and cancer stages. And then at the CPG level, we get a CPG ID, which is an identification address for that ID CPG. So within we get a chromosome and genomic position and gene annotation, genomic region, and methylation beta value. So and importantly, we get the DNA sequence context of the particular CPG site. So this representation is designed to combine the numerical features such as like methylation values and categorical features like gene and the region, along with the sequence-based features, which is an DNA sequence. So by structuring the data in the way, we can able to make the model to learn both biological context and the raw methylation signals. So this is the architecture which I've been working on. So the first, let me explain this whole architecture in the methodology part. So where it starts from the embedding part, where I'm getting the data set which is being pre-processed and converted into JSON. From there, I've been performing a lot of filtrations, lot of filtrations, and then I'll be coming up with features which has been extracted from the whole data. And then I'll be performing an embedding model where I embed both numerical features and the categorical features along the sequence-based data. And then I'll be feeding to the model. Once a model has been done, and then I'll be doing the prediction. So I'll deep dive into about this architecture in the explanation part. So first, here the first step of the methodology is data preprocessing and the feature engineering. So which is extremely important because DNA methylation data is highly dimensional and each contains hundreds and thousands of CPG sites. So usually all features directly would be lead to the high complexity cost and overfitting and reduced model performance. So we address this by designing as a three-tier feature extraction selection strategy maybe. So which each site filters of the data from the different perspective. So first is that biological filtering where we remove CPG that shows variance across the samples because this sites remains almost constant and do not contribute to the distinction between the cancer and the normal. So in addition, we pose a threshold of on the difference of the methylation levels between the cancer and the normal samples. So this ensures that we retain only the CPG sites, the data, the one that only shows the biological meaningful changes between the cancer and the normal. So the second stage is like we have statistical filtering in this filtering. The mutual information is measured based on the dependency between the CPG features and the target level. So including the non-linear relationships. So this is important because the methylation patterns often exhibits a complex and non-linear behavior, so which cannot be captured by the simple correlation. So using this, we select the top 50,000 CPG sites. So that's per sample. So that are most informative for the classification. So this stage ensures that the statistical relevance of the CBG rate along for the target level, whether it is a cancer or normal. So the final stage is like we apply the predictive filtering using a random forest model. So this random forest model provides us a feature importance across score based on how much the each data contribute to the prediction. So from this, we further reduce the feature set to the 15,000, which are most predictive and can be useful in an effective way in order to distinguish between the cancer and the normal. So this steps ensures that the selected features are not only biologically statistic and biologically important or statistically they are statistically relevant, but also directly useful to the predictions also. So overall, this three-stage pipeline ensures that the final feature set is like has a meaningful, biological meaningful and statistically significant, and then predictively strong. So then now we have after this, I mean after the feature selection, the next step is to incorporate the biological structure of the data into the model. So this important because the CPGs are not isolated; they are existing within the genomic environment, and their behavior depends on the neighboring regions and sequence context. So first, we have the model like spatial relationship between we model the spatial relationship between the CPG sites, where we sort the selected CPG site based on the chromosome and the genomic position of that particular data, so that the nearby CPG sites, I mean nearby data, are placed together. So then we can use that sliding window approach, so that we can compute the neighboring mean and neighboring variance of that particular gene region. So this feature captures the local co-methylation patterns, which are biologically important because nearby CPGs are often show the correlation behavior among them. So next we have that we have to incorporate the functional genomic information for each CPG site. So we compute its distance to the nearest transcription site TCS, where it's a transcription site start site, which is an important which plays an important role in the DNA transcription, which takes place in our body. So this CPGs, whichever near to the region, have a strong influence on the gene regulations. So we also encode the categorical information such as like gene symbol, chromosome, and genomic region. This provides us the biological context to each. So the most important step, the most important step is incorporating the DNA sequence context. So for each CPG set, we extract the surrounding DNA sequence, which is an ATGCA. So and passing through the pre-trained model called an nucleotide transformer. So this is a pre-trained transformer which has been trained on a wide number of human genome. So it has the information. It's been trained on information about the human DNA. So this model generates an embeddings of 1280 dimension, which captures the DNA grammar and the regulatory patterns and sequence level biological signals. So this is very powerful because it brings the foundation model knowledge into our model. So we here now, this is the proposed model, which is an biologically structured transformer. So designed in order to integrate the methylation signals with the genomic information. the geometry as well as the sequence context in an unified manner. So at the input level, CPGs are represented as using a combination of numerical, categorical, and sequence based features. So we begin with the processing like the methylation signals, which include the beta value and the beta state, and the neighborhood statistics such as like mean and variance. So they are passed through the multi-layer perceptron layer, so where the lower dimension input is projected into a higher dimension space, so allowing the model to capture the non-linear epigenetic patterns effectively. So in parallel, we encode that biological context features such as like gene, chromosomes, genomic region, set as using a learnable embeddings. So these embeddings are aligned to the common dimensional space and combined to form an unified context representation. So ensuring that each CPG carries a functional biological meaning. To further enrich the representation, we incorporate DNA sequence context data. So for each CPG set, the surrounding sequence is passed through the pre-trained model, as I told you before. And then the CLS token, and we get a class token embeddings, which capture the sequence level semantics. It's extracted, and then it's been extracted and projected into the model space. So these components like signal, context, and position information, sequence embeddings are then integrated using a context-aware gating mechanism. So specifically, we can tell is like it's a sigmoid-based gate. So it is computed, from the combined context and sequence features, and this gate is dynamically scales the methylation signals using the formulation that I have been mentioned here, where signal is multiplied by the 1 plus of the gain. So this allows the model to amplify the biological importance of the CPG while supporting the less relevant ones. For modeling interaction, instead of applying the full self-attention, we use a localized attention where each CPG sign attends only to its nearby neighbors within the fixed window. So this reduces the computational complexity from the quadratic to the linear with respect to the sequence length. So while still capturing the local co-methylation patterns, so which leads us to have a meaningful, biologically meaningful day, analysis. So after capturing the local dependencies, the representations are passed through a lightweight transformer encoder model. So which aggregates this higher level interaction across the CB sites, so that we can have a global interaction among the windowed CPG sites, so that we can able to get the long range dependency. So finally, we apply the global max pooling to extract the most dominant feature, followed with the dropout regularization and fully connected layer to produce the final classification output. So overall, this architecture enables the multimodal fusion, efficiently having the interaction, and then biologically uh, informed feature weighting within the scalable transformer framework. So for training the model, we used a multi-coherent setup where the model was trained on a GEO dataset and evaluated on other dataset, some other data set, which is a TGCA dataset. So this setup is important because it allows the test to model ability to generalize across the dataset distribution, not only relying on a particular dataset. As we are using training on one dataset and testing, evaluating with the TGCA dataset. So to handle the class imbalance between the cancer and the normal data, we use a combination of weighted random sampling and weighted cross entropy loss function. So in order to ensure that the model does not become biased towards the majority class, which is in cancer. So the model was trained using an Adam W optimizer with a learning rate of 10 power 3 into 10 power minus 5. So along with the cosine annealing schedule in order to make sure that it has a stable convergence to the minimal. So during the training, the model has been converged relatively quickly. So after where it achieving the approximately like around 89.1% of validation accuracy within the 5 epoch of data, where each epoch is been batched into 441 batches. So additionally to the performance, we also focused on interpretability. So we applied a gradient-based analysis where the gradients of output with respect to the input features are computed. By analyzing those magnitude of this gradients, we were able to identify which CPG sites are contributed most to the prediction. So using this approach, the model was able to highlight the several biologically relevant genes, including the genes which are mentioned here, which is an HOXA7, HOXA5, and PAX2. So these genes are actually known to be associated with the lung cancer, and it's been proven biologically, clinically. So which and we are getting those same genes as the top most influence gene in our lung cancer prediction. So which validates the model. It's not only learning the accurate, and it also learning the biological insights of the classification. So, while the current model shows a strong performance, there are still we are planning to have an improvement in the model. So, one important extension is like masked CPG pre-training, where the model can be trained in a self-supervised manner by masking the certain CPG sites, learning to predict them. So, this would allow the model to learn the underlying patterns more effectively, similar to the language models. So another direction is that incorporate domain adaptation techniques such as like domain adversarial neural network. So which can help to reduce the distribution difference between the data sets like TGCA and GEO. So improving the model generalization capability among across the data set of any type. So we can also explore the hierarchical gene level modeling where CPG sites, which is data sites, are grouped based on the genes, and then attention mechanism are used to capture the gene level relationships. So making the model to be more biologically structured both and towards clinically. So in addition, the model can be extended to the multitask learning framework. So where it predicts not only the cancer presence but also the cancer stage. So enabling the disease progression analysis also. So finally, the structure can be scaled to an multi cancer classification, not only relying on one subtype. We can go for another cancer like pan cancer, like where the model learns shared methylation pattern across the cancer types, improving its broader clinical applicability. So these are my references. So thank you.